Hi, hello all, welcome to my channel. Today we are discussing about the topic blood groups and the blood typing. So the blood groups are categories that based on the antigens present on the RBC. So the blood typing is crucial for the blood transfusions, organ transplants and pregnancy management to prevent the adverse reactions. So understanding this concept is essential for the safe blood transfusions and the medical procedures. All of you curious about how the blood type is determined. So blood type is determined by the presence or absence of the antigens on the surface of the red blood cells as well as the antibodies in the plasma or the fluid. Antigens are the substance, usually protein in nature, that stimulates the formation of the antibodies. So the antigens are found on the surface of the red blood cells or the RBC. The image which shows the RBC cell having the antigen and antibody is present in the plasma. So the antibody is the protein that produced by the immune system in response to the presence of the antigen. Antibodies are Y-shaped proteins that formed within the blood which react with the antigens. They are found in the blood plasma. The image depicts the interaction between the antigens and the antibodies. So the antigens are yellow. These are the foreign molecules or the parts of the pathogen that elicit an immune response in the body. So each antigen has a unique shape that fits with a specific antibody. So the orange color, which is a marker molecule, this can be a part of the antigen and or associated with it, so which used to identify or tag the antigen. So the then blue color, which is antibodies, these are the Y-shaped proteins that produced by the immune system to identify and neutralize the antigens. So each antibody has a unique region that the arms of the Y that bind specifically to an antigen with a complementary shape. So the image shows several antigens with different shapes. Each antigen has a specific antibody so that matches its shape. So when an antibody binds to its corresponding antigen, it can neutralize the pathogen or mark it for the destruction by other immune cells. So this specificity is crucial for the immune system. The ABO system, it was given by the Karl Landsteiner in 1900. So it states that if an agglutinogen is present on the RBC of an individual, the corresponding agglutinin must be absent in the plasma of that individual and vice versa. The Landsteiner law says if an antigen is present on the red cell membrane of an individual, the corresponding antibody will be absent in the plasma. Or if an antigen is absent on the red cell membrane of an individual, the corresponding antibody present in the plasma. The ABO system, A and B are antigen glycoproteins that found on the surface of person's RBC, and O indicates no antigen present. You know how many different blood types are present in ABO system, and which are they? There are 4 different blood types. They are A, B, AB and O type. There are 4 blood groups. In blood group A, the A antigen present on the surface of RBC and antibody B is present in blood plasma. And in blood group B, the antigen B is present on the surface of RBC and antibody A is present on blood plasma. In blood group AB, both the antigens A and B present on the surface of RBC and no antibodies, that is A and B antibody, are absent in the blood plasma. In blood group O, both the AB antigens are absent, but the AB antibodies are present on the blood plasma. So here is a clear table. In group A, antibody B is present, antigen A present. But in group B, antibody A present, antigen B present. And group AB, there are no antibodies present, but antigens A and B are present. And group O, the antibodies A and B both are present and the antigens are absent. You know which blood type is universal donor and which blood type is the universal recipient. Let me know about this. A person having a blood group A positive, which gives for the A positive and AB positive persons, and a person for A positive blood type, which receives the blood from A positive, A negative, O positive, O negative. So this type, this table is arranged, and the universal donor is O negative and the universal acceptor is AB positive, who can receive blood from all the blood grouped persons. This table tells the how the blood group can be crossing. If the both the parents blood group and the child's blood group, if parents having like if male having O and the female having O group, then the child's group becomes O. If one having O and other having A, then the child's blood group become O or A. And if male having O group, then the female having B group, then the child's blood group become O or B. So this way the child's blood group can be determined. Then RH factor or the rhesus factor. So besides the ABO system, a person can either be RH positive or be RH negative. So most of the people are RH positive. So the RH factor is another antigen that found on the surface of the red blood cells. So it can be a problem if a woman is pregnant and she is RH negative and the fetus is RH positive. But normally a mother and her baby have separate blood system, but sometimes when the baby blood may enter the mother system, it may affect. And in pregnancy, if the mother is RH negative and is exposed to her baby's RH positive blood, the mother's body produces antibodies against the baby's red blood cells. But this is not a problem during the first pregnancy, because in that time antibodies haven't built up. But during other pregnancies, the mother is given a drug to suppress her reaction to the baby's RH factor. Let me know about the transfusions. So blood transfusions is the process the transferring the blood or blood based products from one person into the circulatory system of the another person. Sometimes mixing blood from two individuals can lead to a blood clumping or even death also. So this occurs when the blood of the donor and the blood of the recipient are not compatible. So blood groups and typing is very important here. When we receive blood that doesn't match our own, our body produces an immune response. So this immune response is similar to one produced when our cells encounter a pathogen. So when a pathogen with unknown antigens invades our body, an immune response which is stimulated, then the antibodies attach to the unknown antigens and deliver the pathogen to the macrophages, which destroy the pathogen. Then all may curious about how the blood typing done, so which is used to determine the blood groups of an individual. So involves the mixing the blood with three different reagents that have the A antibodies, B antibodies and the RH antibodies. In blood typing, the main point is agglutination or the clumping, that indicates that the blood has reacted with the antibody reagent. For example, in blood type A, that will clump when placed in antibody A reagent. So another example, blood type O will not clump in any reagent since it has no antigens. So this image explains the how to interpret the blood typing results using the agglutination reactions with anti-A, anti-B, anti-D, that is RH antibodies. So blood type indicates the ABO and RH type, that is positive or negative. In column anti-A, anti-B and anti-D and the control. So anti-A that test for the presence of A antigen, and anti-B the test for the presence of B antigen, anti-D that test for the presence of RH antigen, and the control that ensures the test is working properly, and they should show no agglutination. So in O positive, anti-A, anti-B and the control that having no agglutination, and in anti-D that agglutination occurs, and in O negative, anti-A, anti-B, anti-D and control that or absence in the agglutination, and in A positive the anti-A and anti-D have agglutination, and anti-B and control they have no agglutination, and in in A negative anti-A have only the agglutination, and in B positive only the anti-B and anti-D or agglutinate, because here anti-D that is a RH, where RH present the blood type becomes positive. In B negative only the anti-B have agglutinate, and in AB positive the agglutination occurs in anti-A, anti-B, anti-D, and AB negative anti-A, anti-B or agglutinate, and that is invalid. Invalid means that any combination when the control shows the agglutination or where results do not match a valid type. So the agglutination indicates the presence of the corresponding antigen. For example, if the sample agglutinates with anti-A but not with the anti-B and does agglutinate with anti-D, the blood type is A positive. So if it agglutinates with neither anti-A nor anti-B but does agglutinate with the anti-D, the blood type becomes O positive. This is O negative blood in A and B type in RH. The agglutination is absent, so the O negative blood is a universal donor. It is AB positive, that is a universal recipient, because agglutination occurs in anti-A, anti-B and anti-D. It is O positive blood. The most common type is O positive blood. The agglutination occurs only in the anti-D, because in O positive the anti-A, anti-B are absent. In A positive the only anti-A and the anti-D have agglutination. This is A positive blood. In B positive the anti-B and anti-D are agglutinate. In the agglutination absent in the anti-A, it is a B positive blood. So this image illustrates the compatibility of the blood types for the donation, and the arrow shows the direction of the compatibility, that indicating the which blood types can donate to which other blood types. So this compatibility is crucial for the safe blood transfusions. So in O type blood, that is a universal donor, O type blood can be donated to all the other blood types, that is A, B, AB and O. A type blood can donate to only A and AB blood types, and B type blood can donate to B and AB blood types. AB type blood is a universal recipient, that AB type blood can receive blood from all other blood types, that is A, B, AB and O. Here see what's your blood type. So the most common blood type is O positive. The 38% people have O positive blood group, and very rare blood group is AB negative, that is only 1%. It is a revision purpose, that is ABO and RH systems, that blood type determined by the antigens that located on the red blood cells and the antibodies that located in the plasma. There are 4 different types of the blood, that is A, B, AB and O, and RH positive or RH negative. The transfusion dependent on the immune response, that antibodies present or absent. In blood type A, antigen A present. In blood type B, antigen B present. In blood type AB, the both antigens are present. In blood type O, they are absent in the antigens. I hope you people understand it very well. Thank you all. Please like, share and subscribe my channel for getting a more videos about biology topics.